Welcome everyone. Viruses, being incredibly small, often go unseen by the naked eye. Powerful microscopic techniques like immunofluorescence and electron microscopy allow us not only to see these tiny invaders but also to identify them.
Modern virology research has become as crucial now as ever before due to the global COVID-19 pandemic. There are many powerful technologies and assays that virologists can apply to their research into the structure and function of viruses. One essential and often overlooked aspect of understanding the behavior of viruses is the ability to visualize their effects on host cells. As such, immunofluorescence represents a precise and powerful tool that can be used to observe viral infections and their effects on host cells. Immunofluorescence is a histochemical staining technique used for demonstrating the presence of antibodies bound to antigens in tissues or serum. Immunofluorescence relies on the use of antibodies chemically labeled with fluorescent dyes to visualize molecules under a light microscope. In immunofluorescence, antigens, antibodies, and their complexes are viewed under a fluorescent microscope. The combination of antibody with its specific antigen does not lead to a visible change, and thus a readily identifiable label, namely a fluorochrome, must be irreversibly bound to the antibody so that its localization can be recognized. Fluorophores, also called fluorochromes, is a chemical molecule which is used to label specific structures for microscopic analysis. The most commonly used fluorophores are fluorescein isothiocyanate and tetramethylrhodamine isothiocyanate. Now, let us look into the principle of fluorescence emitted by these fluorophores or fluorochromes. Fluorescent materials give off light because of their atomic structure. Electrons are arranged in discrete energy levels. When an electron absorbs energy from a photon of light, it becomes excited and jumps to a higher, less stable energy level. The excited state has a half-life of less than 10 seconds. The electron loses a small amount of energy as heat, and the remainder is given off in the form of a photon. The emitted light has lower energy than the absorbed light and hence higher wavelength. The emitted light is almost invariably in the visible spectrum. The fluorochrome markers are detected with a fluorescence microscope equipped with a mercury vapor or xenon light source and appropriate filter, the excitation and barrier filters. The exciter filter serves to shed the light of necessary wavelengths on the examined slide, while the barrier filter stops the exciting photons, letting through only the fluorescent light. Since antibodies play a central role here, it is worth taking a closer look at them before diving into the technical aspects of immunofluorescence. Antibodies are Y-shaped molecules, proteins formed by four chains, the two so-called heavy chains and two light chains. The antibody stem and part of arms are identical for all antibodies of one class and are therefore called the constant region. The outermost part of the arms, however, differs from antibody to antibody and it is called the variable region. The variable region of the heavy chain and the variable region of the light chain together form the antigen binding domain. It is this part of the antibody which recognizes and binds the target structure epitope, hence determining the antibody specificity. Differences between monoclonal and polyclonal antibodies. Monoclonal antibodies are produced by the clones of a single B cell. They are completely identical and recognize and bind the same epitope. Due to their specificity, they display very low cross-reactivity, but are comparably insensitive as every target protein is bound by only one antibody. Polyclonal antibodies originate from a pool of B cells. They recognize a variety of epitopes on target protein which increases sensitivity but might bind to epitopes of other proteins than the one of interest. Overview of the immunofluorescence procedure. First, fixation. Chemical fixative like formaldehyde helps preserve the structure and immobilize the proteins while fixing the cells and tissues. The method selected should preserve sample morphology with minimal impact to the target epitope. There is no one-size-fits-all method. The method selected depends on the target and sample type. Step 2. Sample preparation. In case of cells, culture them on glass cover slips or chamber slides. While using tissues, prepare tissue sections by fixing, embedding, and sectioning the tissue. Again, the methods depend on the target sample type. Examples of methods used include permeabilization. Permeabilization is often necessary to allow antibodies to enter the cells or tissues. This step involves treating the sample with the help of detergents or other agents. Step 3 is blocking. This step helps reduce the non-specific binding of antibodies. Here, the sample is intubated with a blocking solution like bovine serum albumin or normal serum. Step 4. Incubation with the primary antibody. Here, the primary antibody is applied to the tissue section. This antibody is specific to the target protein in the sample. The antibody binds to the target protein within the sample. This is followed by washing. The use of buffer solution helps in removing excess primary antibodies. Secondary antibody incubation. The antibody conjugated to a fluorescent dye is used in the step. This secondary antibody recognizes the primary antibody and binds to it. This is followed by washing again to remove excess of secondary antibodies. Finally, mounting. A mounting medium is used with anti-fading agents for mounting the sample to preserve the fluorescent signal. Imaging. The fluorescent microscope with appropriate filters examines the sample. This helps visualize the fluorescent signal emitted by the secondary antibody bound to the target protein. Different fluorophores emit different colors of light, allowing for multicolor imaging. Now, let us look into the different types of immunofluorescence. Direct immunofluorescence. In the direct method, the fluorophore label is conjugated directly to the primary antibody that will be reacting with the target epitope in the tissue. Indirect immunofluorescence. The indirect method involves a two-step incubation process. The primary antibody binds to the target epitope. Step 2. A fluorophore tagged secondary antibody recognizes and binds to the primary antibody. Although the direct method is quicker, the indirect method is more widely employed for its high sensitivity, signal amplification, and its ability to detect several targets in the same sample. Amplification of signal with polyclonal secondary antibodies labeled with the fluorophore. In choosing proper antibodies, several criteria must be taken into consideration. To prevent secondary antibody from cross-reacting with endogenous immunoglobulins in the tissue sample, the primary antibody should be derived from a different species than that of the sample. Consequently, the secondary antibody must be against the host species of the primary antibody. Secondary antibodies can be modified and conjugated in multiple ways for purposes of visualization and signal amplification. Commonly, secondary antibodies are conjugated with fluorescent labels which emit fluorescence upon photoexcitation. Enzymatic labels can also be conjugated to react with chromogen substrates for colorimetric analysis. For greater signal amplification, polyclonal or biotinylated secondary antibody can be employed. For each primary antibody, polyclonal secondary antibodies that are able to recognize multiple epitopes to increase binding and signal levels can be employed. Amplification of signal by secondary antibody conjugated with multiple fluorophore protein complexes. Similarly, multiple fluorochrome protein evident or streptavidin complexes can bind to a single biotinylated secondary antibody to increase the signal levels. Amplification of signal with polyclonal secondary antibodies conjugated with multiple fluorophore protein complexes. The immunofluorescence method has inherent advantages with regards to signal amplification, targeting specificity, resolution, and analytical capabilities. The immunofluorescence method can be further improved for greater amplification of the signal with the use of polyclonal antibodies in conjugation with enzyme complexes. One such example is the fluorophore labeled streptavidin biotin complex. Being polyclonal, multiple biotinylated secondary antibodies are able to recognize and bind to a single primary antibody. In turn, binding of greater numbers of fluorophore labeled streptavidin per primary antibody greatly amplifies the fluorescence signal relative to a simple direct immunofluorescence method. In addition, immunofluorescence methods allow the possibility of multiplexing. Complement fixation immunofluorescence technique is a very sensitive three-step serological technique. Through this method, very small quantities of circulating antibodies are detected by means of their high affinity to fix complement. The complement can be demonstrated using the FITC labeled anti-complement antibody. The technique is more sensitive than routine immunofluorescence due to the amplification achieved through binding of more than one molecule of complement to each immunoglobulin and the subsequent visualization of the multiple molecules of complement. Now let us go through a brief overview of how immunofluorescence helps in virology research. It helps study the mechanism of viral infection in cells. Immunofluorescence assays help study how viruses bind to cells, release genetic material, and assemble new particles. Example to study the mechanism of infection of SARS-CoV-2. It helps study the effects of viral infections on cell function. Immunofluorescence helps visualize the cellular response to viral infection. Examples: it helps study stress granule formation during rabies infection or repair mechanisms in influenza infected cells. It helps monitor the treatment effects. Immunofluorescence can be used to test antiviral agents, to localize them, and to study their behavior within cells. It helps to analyze if the antibody cocktail shows any binding to the viruses. It helps monitor treatment effects on viral particle presence and localization. Here is a super resolution microscopic image of mammalian cells infected with Zika virus and labeled for indirect immunofluorescence. This represents a mock infected viro cells, which is the control cells which are treated with all reagents but not the virus. These are the Zika virus infected cells. Here we can see the Zika virus in the perinuclear region. Advantages of the immunofluorescence technique. The immunofluorescence technique is simple and reproducible. It is a short procedure which can be completed within a time period of 1 to 3 hours. It shows high sensitivity and it has substantial prognostic value. Limitations of immunofluorescence techniques. 1. Photobleaching. This is the permanent loss of fluorophore's ability to emit light upon exposure. During immunofluorescence, the high-intensity light used for visualization can damage the fluorescent molecules, fluorophores, attached to the antibodies. This leads to a decrease in signal intensity over time, making it difficult to observe the target antigen clearly, especially for longer observations. Autofluorescence. This is the emission of unwanted background fluorescence from the sample itself, not from the target antigen. Cellular components like collagen, elastin, and certain lipofuscin pigments can emit natural fluorescence that interferes with the specific signal from the labeled antibodies. This can make it challenging to distinguish the target antigen from the background noise. Fluorescence overlap. This occurs when multiple fluorophores used in the experiment emit light at similar wavelengths. If these fluorophores are not efficiently separated, their signals can overlap, making it difficult to distinguish between the different antigens being visualized with different colored dyes. Non-specific fluorescence. This arises from non-specific binding of antibodies or fluorophore conjugated molecules to irrelevant structures in the sample. This can happen due to the inadequate washing steps or using antibodies with low target specificity. Non-specific fluorescence creates background noise and reduces the clarity of the specific signal. Multimerization and effects on target molecules. Antibodies can sometimes form clusters, multimers, around the target antigen. This can potentially hinder the proper binding of other antibodies or probes, affecting the overall staining intensity and potentially masking some target molecules. Additionally, the antibody binding process itself might slightly alter the conformation or accessibility of the target molecule, impacting its true representation in the sample. These limitations can be mitigated through various strategies, minimizing light exposure during observation, using specific quenching agents to reduce autofluorescence, selecting fluorophores with distinct emission spectra, optimizing washing steps and antibody concentrations, employing blocking agents to minimize non-specific binding. By being aware of these limitations and implementing appropriate controls, researchers can obtain more accurate and reliable results from the immunofluorescence experiment. Electron microscopy is an efficient tool to demonstrate viruses for diagnostic purposes, for structural analysis, and for studying virus-cell interactions. Principles of electron microscope. Two kinds of electron microscopic technologies have been developed: transmission electron microscopy (TEM) and scanning electron microscopy (SEM). TEM is a microscopic technique in which a beam of electrons is transmitted through the ultra-thin specimen, interacting with it as the beam passes through. TEM relies on negative staining of purified virus particles with an electron-dense material such as uranyl acetate or phosphotungstate, and images formed from the interaction of electrons transmitted through the specimen. The image is magnified and focused onto a layer of photographic film. What can be learned from TEM images of virus particles? TEM reveals the overall morphology, whether viruses are spherical or elongated particles, and whether they are naked or enveloped. In addition, even symmetric pattern, that is, the T number of an icosahedral capsid, could be obtained from a higher resolution image. On the other hand, SEM is a type of an electron microscopic technique that produces images of a sample by scanning it with a focused beam of electrons. The electrons interact with the atoms in the sample, producing various signals that can be detected and that contains information about the topography and composition of the sample surface. Here, in these images, the principle of TEM and SEM is illustrated for comparison. Electron beam lines are indicated by the red line. TEM generates the image by electrons that are transmitted from the specimen, while SEM generates image by electrons that diffract from the specimen. Here we can see representative electron microscopic images of virus particles. The first one is a transmission electron micrograph of rotavirus particles. The second one is a scanning electron micrographic image of HIV particles, these green particles, budding from an infected T lymphocyte. Most biological materials show little contrast with their surroundings unless they are stained. In the case of light microscopy, contrast can be enhanced by using colorful stains which selectively absorb certain wavelengths. The electrons in the electron microscope are absorbed very little by biological material, and contrast is obtained mainly by electron scattering. To heighten the contrast between viruses and the background, use is made of electron-dense stains. These are usually compounds of heavy metals of high atomic number that serve to scatter the electrons from regions covered with the stain. If virus particles are coated with the stain, positive staining, fine detail may be obscured. Negative staining overcomes this problem by staining the background and leaving the virus relatively untouched. The negative stain is molded round the virus particle, outlining its structure, and is also able to penetrate between small surface projections and to delineate them. If there are cavities within the virus particles that are accessible to the stain, these will be revealed and some of the internal structure of the virus may be disclosed. Here is the comparison of herpes virus appearance after positive and negative staining under an electron microscope. Immunoelectron microscopy can be defined as any technique that uses antibodies or molecules that interact with antibodies. For example, protein A or protein G in conjunction with electron microscopy to localize antigens or antibodies in cells and tissues. A sample containing the virus is prepared and placed on a grid. Antibodies specific to the virus of interest linked to an electron-dense marker like gold particles are added to the sample. The antibodies bind to the virus particles, antigens. The sample is then washed to remove any unbound antibodies. Why use immunoelectron microscopy (IEM) for viruses? Specificity. IEM is more specific than traditional electron microscopy, which can only visualize the general structure of a virus. IEM can differentiate between different viruses even if they have similar morphologies. Sensitivity. IEM can detect very small amounts of virus, making it a valuable tool for diagnosing viral infections. Localization. IEM can be used to determine the location of viruses within cells or tissues. This information can be helpful for understanding how viruses infect cells and cause disease. The grid is examined under an electron microscope. The electron-dense markers make the virus particles visible, allowing researchers to identify their location and morphology, shape, and size. This is an immunoelectron microscopic image of wild-type influenza virus. Immunogold labeling. Immunogold electron microscopy is a classic high resolution method for the selective localization of macromolecules in the context of cells and subcellular structures. Specific antibodies affix small particles of colloidal gold which are easily visible in the electron microscope to the macromolecule of interest. There are different immunogold labeling techniques. In the post-embedding immunogold labeling technique, the biological material is first fixed, dehydrated, and embedded in a resin, and the antibody reactions are done on the sectioned material. In pre-embedding immunogold labeling technique, the antibody reactions are carried out prior to fixation, dehydration, and resin embedding of the biological specimen. The whole mount immunogold labeling technique does not involve resin embedding at all. The material is applied to an electron microscopy grid and the antibody reactions are carried out on the grid. What is cryo-electron microscopy? Cryo-electron microscopy is an advanced imaging technique used to elucidate the 3D structure of biological molecules and complexes at near atomic resolution. In this technique, the sample is rapidly frozen to temperatures below 150 degrees centigrade, trapping it in vitreous ice. The sample is then imaged from different angles using an electron microscope where a beam of electrons passes through the specimen, resulting in a set of two-dimensional projections. Advanced computational algorithms are then used to reconstruct a 3D model of the sample from these projections. The cryo-electron microscope workflow begins with the preparation of the sample, typically a purified protein or complex in solution. The sample solution is placed onto a specialized grid and then it is rapidly frozen by plunging into a cryogenic fluid such as liquid ethane. This transforms the water into amorphous ice, causing vitrification of the sample and preserving the native structure of proteins. The vitrification procedure can be performed following different methodologies, some of which are optimized to capture short-lived molecular states. Next, the vitrified sample is placed onto the grid and transferred to the cryo-electron microscope where it is maintained at extremely low temperatures to preserve sample integrity. The microscope is calibrated and aligned and data acquisition begins. An electron beam is directed at the sample, producing a series of 2D projection images from different angles. These 2D images are processed to correct for artifacts and improve the signal to noise ratio. Individual particles within the images are identified through particle picking and large data set of particles is collected. Computational algorithms are then employed to reconstruct a 3D density map from the particle images. The resulting 3D map provides a representation of electron scattering intensities of the sample, allowing scientists to gain insights into its structure. Atomic models are fitted into density map and refined iteratively to represent the experimental data accurately. The refined models are validated and analyzed to understand the structural features of the studied molecules. High resolution image obtained by cryo-electron microscope. Cross section and two cut-away views of hepatitis B virus obtained by cryo-electron microscope. HBV virion is composed of a T54 icosahedral capsid with 120 spikes and an outer envelope with projections of envelope glycoproteins. The image analysis of capsid shell yields an interior dodecahedral cage of density, which is ascribed to ordered, mature, double-stranded DNA. Strengths: high near atomic resolution imaging. Sample preservation enables the study of samples in their near native state. It can be applied to a wide range of biological samples, including proteins, viruses, cells, and tissues. It can be applied to study large, flexible complexes and assemblies that are challenging for other techniques. It enables single particle analysis with no size limit or need for crystallization. Limitations of cryo-electron microscopy. It requires certain homogeneity in the particle population to achieve high-resolution determination. It generates large data sets that are computationally demanding. There are resolution limitations and sometimes structural details cannot be well resolved. Larger samples are difficult to study. Cryo-electron microscope equipment is costly and requires expert users. Now let us discuss some MCQs on this topic. Which of the following microscopy techniques provides the highest magnification for visualizing viruses directly? A. Immunofluorescence IF. B. Electron microscopy EF. C. Cryo-electron microscopy cryo-EM. Or D. Immunotransmission electron microscopy immunotem. The answer is C. Cryo-electron microscopy. Immunofluorescence relies on the specific binding of antibodies to dash to visualize viruses in a sample. A. Viral proteins. B. Viral RNA. C. Viral DNA. D. All of the above. The answer is A. Viral proteins. Immunotem is a valuable tool for diagnosing viruses because A. It can differentiate between live and dead viruses. B. Provides high-resolution images of virus-antibody complex. C. Is a more sensitive technique compared to immunofluorescence for all viruses. D. Requires minimal sample preparation compared to other methods. Answer is B. Provides high-resolution images of virus-antibody complex. When interpreting a virus diagnostic test result, it is important to consider A. The specific test used and its limitations. B. The patient's symptoms and medical history. C. Both A and B. Answer is C. Both A and B. Cryo-electron microscopy requires flash freezing the sample in what to preserve the virus in its near native state? A. Water. B. Liquid nitrogen. C. Carbon dioxide. D. Oxygen. Answer is B. Liquid nitrogen. Thank you for listening.